Assalamu alaikum. Good evening, everybody. My name is Professor Ismail Ahmed, Department of Microbiology, Aliko Dangote University of Science and Technology, Wudil. Today, we are going to discuss on steps for writing a research report. That is, when you finish conducting your research, then you have to be writing the report. It is advisable that when you are conducting the research, you're supposed to combine it together with the writing of the research report. It's not to say when you finish completely, then you now come and begin to write the report. Already, it's assumed that you have conducted a presentation for proposal presentation. You propose, you present, and then it is approved. So as you are doing your work, you continue to develop your work. You continue to write your report. Don't wait till the end of your research, then you begin to write your report. So the steps are how you write it is it contains some preliminary pages, then chapter 1, chapter 2, chapter 3, chapter 4 for undergraduate project. But there is chapter 5 for master's dissertation, and there is chapter 6 for PhD thesis. So depending on the regulation of your university, then you now try as much as possible to abide by the regulations of the university. So, but these are the general steps. So the step will include the preliminary pages. So what is in the preliminary pages? There is what you call a cover page. It's designed in such a way that it will now serve as a information which you see, just maybe the title of the course, then it is by your name of the student, and the year, the date. So that will only be in the cover page. Then there is what you call a flyer page, whereby you stick the flyer for the award of the degree. But nowadays, because people normally used to tear it, so we allow the flyer page to be sticked on the hard cover bound cover. So we assume that the flyer page should be at the back of the hard bound copy. So after the cover page, sometimes the cover page is 1 or 2. So you can provide 1 or 2 cover pages depending on how normally the guideline stated. So the preliminary pages are numbered in lower case, Roman numbering. So page 1 should be title of the title page. That is which will contain your title, your name of supervisors. Then the write-up is it project or for masters or for PhD, for the award of higher of the degree, and it's also department submitted to department. You mention your department, and then you mention the faculty, the School of Postgraduate Studies, and in partial fulfillment for the requirements of the award of degree. Then you mention the degree, and then if there is an option for the degree, you can mention the option. So that is title page. Then you come to declaration page. Declaration page, that is declaration. It's a page title called declaration. The heading will be declaration, and then this is where you're supposed to declare that you have conducted the work, and you didn't copy it from anybody. You just conduct the project or the research, and then according to the guideline of the university, then you sign the page, only use the student. Then certification page, number 3, page 3 will be certification page, whereby your supervisors will certify it. So it depends on how many supervisors you have, whether sometimes we only have 1 supervisor for undergraduate project, 2 supervisors for masters, and 3 supervisors for PhD. So this is where they will certify. Yes, you make a statement. You have conducted the work under the strict supervision and according to the guideline of your institution. Then your supervisors will certify. So some people match certification page. It's supposed to be certification page or approval page, where all the other people will sign, the head of department, the head of faculty. But it's advisable to have a page 4 called approval page. Approval page, whereby whereby those who are supposed to approve the work, including your supervisors, including the PG coordinator, including the head of department, especially also including the external examiner who come for your Viva, is supposed to approve that page. So depending on how the arrangement, the guideline will state it, your institution guideline will state how you arrange them. Sometimes we started with the external examiner who came and approved the result, then followed by maybe your internal examiner, and then the coordinator, the PG coordinator, depending on how your university say you have arranged it. Then there is what we call dedication. It's a page called dedication, which is supposed to be page 5, and the dedication page is the one that you dedicate to the work which you dedicate the work to. Maybe you are dedicating to your late mother or to your late father, or you are dedicated to your community people and the rest. So please don't do too much English there. Just go ahead and dedicate your work to whom you want to dedicate the work to. Some people, they wanted to do something. Maybe they wanted to dedicate to 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 God. Sometimes it's not, because this is not what you just dedicate to God, but try as much as you dedicate your work to somebody. Maybe some people will dedicate to Jesus Christ. I think you should just dedicate your work to whom you desire to work, because you are not writing something strictly religious for you to say you dedicated this work to God. Because you are writing project probably on some other issues which somebody can say is not correct or is correct. So please, I am trying to advise, don't dedicate to God Almighty, but just try to find somebody in your mind, in your heart, that you can dedicate this work to, and just go straight to the point and dedicate your work to him. Then another page called acknowledgement page, which is supposed to be page 6, acknowledgement page. So all in acknowledgement, that is why you acknowledge the God Almighty, you acknowledge your parent, you acknowledge your supervisors, you acknowledge teachers that teach you, that taught you during the coursework, and then you acknowledge all those who participate in one way or the other, including those who give you smile, who make you to smile while you are annoyed during the process of the study. So including the people in the lab, including other students that help you one or the other. So then after acknowledgement, then there is what you call table of content, whereby you do your table of your content, what you see inside. Maybe then other list of content, maybe list of tables, list of plate, list of abbreviation, and so many others. And then you come to very important things at the last, just before entering chapter 1. That is abstract. Abstract is very important. That is trying to showcase what is the inside, what you are going to see inside the work. So there is what you call extended abstracting. What do you mean by extended abstraction? You start with your aim, and sometimes not necessarily aim, but you start with introducing the title. Try to shed more light, make an introduction whereby you show more light on the title. Then from there you now state your aim. After stating the aim, then you state the objectives. Then you study the methodology, the method, some of the important method you use. Then you state the result clearly according to your own objectives. Try to make sure all the objectives have been highlighted, the result of each objective has come out. Then you try to conclude and summarize. You try to conclude and summarize. You try to summarize, and then you conclude. So that is all for preliminary phases. Now chapter 1. Chapter 1 is introduction. Chapter 1 is introduction. You try to introduce your title. So in the introduction, in the title, you will do what you call you will do what we call keywords from the title, and make sure in the introduction you have addressed all the keywords. All the keywords have been addressed. So you make sure in the introduction you address all the keywords. You can start 1.0, 1.0. You start with 1.0, and then you that is introduction. You can give background of the research. Then you can give also 1.1, 1.2, depending on the content you are trying to address. So depending on the content you are trying to address, you will now you will now you will now make make the chapter 1 very interesting. So in chapter 1 also will include statement of the problem. You state the problem, and then you state the problem. When you state the problem of the research, you have to quantify the problem. That is, by quantification means you have to adding weight to it. That is, if you quote number, you quote percentages. Sometimes percentage, you quote your result in percentage, or your maybe some findings in number. So that is quantification. Then you also give references to the research problem. Then research justification, whereby you show justification of your research. Then aim and objectives. You give aim, or only one aim sometimes, for the academic research, mostly of the degree awarding research. And then you give specific objectives. That means you give specific objectives. Specific objectives are those objectives that are normally it's a work conducted in the field or work conducted in the lab. So you find out there are specific objectives. You can have so many objectives, of course, but please don't give all the objectives. Try to give only specific objectives, those that concern work, and then that will have results. That is what we call an objective, specific objectives. So you see, but you have to look at your title also, because all this writing report is somehow connected with each other. You continue connecting up to the end. So you look at your title, make sure your title, your objective has addressed your title. Don't just give a title and then your objective will look different from title. And please, objectives are specific. It's a work that you have to do either in the field or in the lab, not a comprehensive objective. For example, sometimes people will say they want to detect free balance inside an objective. Free balance is too much for objective, so please take note. Then maybe some other things, research questions, research hypothesis, and so many other things that will come after research. Then chapter 2 is what we call literature review. What we mean by literature review is you review the work of others, the work of previous scientists who work in your direction, relevant to your own field of knowledge. So you see, you begin taking the literature reviews. You begin reviewing this literature, what is this work said. So it's just like this abstract. Try to copy the abstract. Literature review includes the title of this work or the aim of the work, and then which method did they apply, then how did they get to their own result, and then what are they concluding, something like that. That is what we call literature review. So when you are doing a literature review, try to capture the information worldwide. So you can do like deductive, and maybe from head to tail, or from tail to end. You can begin with the local information and then expand it to the worldwide issues, or maybe begin to worldwide issues and extend, then lower it to local issues. So you have to quote literature reviews in all these continents: American continent, Asian continent, Indian part of India, Nigeria, northern Nigeria, something, so that you can have a mixture of what is happening in the world, in the current issue of the world, and your references has to be recent, except if maybe that showing an old something is part of what you want to show, something that this is too old. but you have to be current. So you have to also address the keywords in your title when you are doing literature review. For example, if your work title include susceptibility, you give something on susceptibility. You give literature review on susceptibility. If it include extract of certain plant, then you give what is the previous work of extract of those plant, so that it will now address not only taking one part in your literature review. Then chapter 3 is materials and method. That is the procedures you are going to use. So please take note, in chapter 3, when you say material and method, it's not good to start listing materials. It's not good to start listing your method. You just give, because you are writing a report. Not say you are now trying to teach student or to give a procedure for class to conduct the experiment, but you are writing a report. So since you are writing a report, there is no need for you to begin to list how you conduct or how you prepare the media. That one will be in the appendix, how you prepare the media, how the media is being prepared. But this one, just give us materials and method according to how you carry it. So it depends. Some people may begin with study area or maybe study site, depending on what do you call your is it site or area. You know, there is difference between study area and study site, sampling site. Sampling site is a specific location whereby you go and sample, maybe like soil sampling or food sampling. You are going for specific place to sample that place, while study area, you have decided to work on people in Kaya local government probably. So you have to select some words that you go to this place, that is sampling site. You go there and see. But when you are talking of study area, you are talking of the whole area, the whole area of coverage. So like when you say in Kano State, you cannot do it all in Kano State, but you can take maybe 3, 4, 5 local government and conduct your research there, but still you can call it in Kano State. In Nigeria, for example, you can take 3 states, 4 states, 5 states, and then you conduct your work then later on. But you know, the mixture you have to have a reason, a demarcation, whereby you said, why did you select this area, not area? Because when you say you do in Kano State and you selected Gaya, Udil, and Ajinki, some people will say, why did you select in one direction, one area? But when you have to have a uniform distribution, that you have selected one from this geopolitical zone, one from other zone, one from political zone, something like that. Kano Central, Kano South, Kano East, something like that. So you selected, and you now maybe select maybe 3 or more people from Kano Central, because Kano Central is a bigger area. You can have your own study area. Then study area will include study population, longitude, latitude, or GPRS of the place, and references. So make all issues to do with chapter 3, material and method, must include reference. Most of the place. Study design. How do you expect to design your study? That is study design. How do you expect to design your study? Do you intend to go to sampling every week? And then how do you do it? Do you sample, for example, when you select 10 local government in Kano State? So in this today, you go to this local government. Next tomorrow, you go to this local government. This is how you design. So if you design your studies, please try to abide by it, because if you change the design, that means you can have a very poor result. Because, for example, scientific is systematic way of doing things. Systematically, you are doing it systematically. When you go this week, next week you go again, next week you go again, something like that. So you have to have a pattern. Then we have what we call ethical consideration. So most of the studies now, you have to have what you call ethical consideration, and for ethical consideration, that is for ethic, so that you go to the relevant body and obtain consent. Those that are going to participate in your research, you have to consent them. You design a consent form for the participant, then you give them form to fill. Then already maybe there is an authority, maybe hospital management board, give you permission to maybe go to some hospital and conduct your research there. It's very important. Uh, Sample size calculation. So you have to also, in material and method, calculate your sample size. You have to use formula and calculate sample size, or maybe use a pattern using previous prebalance with a formula and calculate your sample size. You cannot just sit down on your own and just decide that I want to so people to participate in my study, or I want so number of sample. Then sample collection, how you collect sample, inclusion exclusion criteria. So when you are collecting sample, there is a specific sample you want to collect. So try to specify inclusion and exclusion criteria. Then apart from that, then the normal other work will follow. That is analysis, your own analysis. But please don't mention analysis. Don't say analysis. So just mention what you are doing. Are you going to do culture and isolation and identification? So just state how you do culturing, how you do gram staining, how you conduct biochemical test, and then you continue the method according to your own objectives. When you now finish this something, this like a highlight at the beginning. I think everybody needs to study size, study design, ethical, sample size calculation, sample collection. But after sample collection, everybody will now do his own analysis differently. So depending on what you are doing in your analysis, follow your objectives and come arrange your methodology in that order. So for example, if somebody who is doing isolation and identification, so he will do culture, isolation, gram staining, biochemical test. Maybe after biochemical test, you are going to do sensitivity, then you do sensitivity. After sensitivity, then you want to do molecular characterization. So you just show your work in that way, but you have to include all others. Now, results and discussion. Chapter 4, result and discussion. So that is result and discussion. You see, every day some new aspect are now coming out and often up. Like for example, the current situation now, you present your result as a result and discussion. So the result, you have to interpret the result. Then you put it in the table 4.1, maybe as you are given, that is chapter 4, 4.1. So you now put the table. You have already interpreted the table, explain the table to those who are reading your report. Then you now put the table. Then you now discuss the table under the table. After you put the table, when you put the table, then you have to put keywords. If there are some certain things which people want to explain, the table has to be self-explanatory. So you now put the explanation, the discussion under that table. But sometimes, mostly what we are doing is we now put results and discussion, then 4.1 results. Then you interpret all your results, then you put the result table page by page. Then you now do the discussion. You now do the discussion, but depending on the guideline, so you do what is supposed to be done. Follow the guideline. When you are discussing your work, you see, you have to discuss your work, because some people will do a very good job, but when they come to discussion, you will find them that actually it's like they don't know anything. But discussion has to be scientific discussion. You give evidence. For example, now, why did your result is like that? For example, when you have a prevalence, is that prevalence very high or very low? Then what does it mean? When it is high, what does that mean? What do you think is the causes of the high prevalence, or what do you cause is a lower prevalence? So you have to explain, and what does it constitute to have lower prevalence? You give a scientific reason, an explanation of why your result is. Then you now try to compare your result with the previous researchers. This is in accordance with one. Maybe, for example, the reason why your result is high, maybe also somebody get higher result and he has implicated some factors, risk factors. There is association between risk factors and your findings, maybe a disease. So when you find 60 people out of 140, they have typhoid fever. So that result, the result, is it high? The prevalence, is it high or low? So you have to compare with the previous researchers, then you know why you can now, okay, it's now high or it's now low. Then what do you think causes that high prevalence? What do you think? Maybe activities, some factors in the community causes that prevalence. So you mentioned some factors, you implicated some food, and some water bodies and some other things. But you you are the one who carry out the research. So you already go for sampling, go inside the town, you observe in the town. So you know what factor that constitutes that higher prevalence. So that higher prevalence, naturally you know why did they cause it, and then you correlate your findings with the previous researchers, or you disagree with the previous researcher. For example, your result, maybe you will disagree with the research of somebody who conducted the research previously. So it doesn't have to always go together. You can agree or disagree is the findings. So you also have to discuss your result table by table, findings by findings. You also discuss in according to your objectives. So when you all tables, or maybe objective one will have 2 tables or 3 tables. So table 4.1, 4.2, 4.3 will all be objective 1. So you have to finish discussing objective 1, table 4.1, 4.2, 4.3, then you enter 4.4, 4.5, something like that. So discuss your work paragraph by paragraph in accordance with your own chapters, with your own tables. You present your result naturally in tables or figures or chart or plate, something like that. So you try to have different mixture. Don't just continue every time you are presenting your result is table, 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 table. No, try to have a mixture. Some are tables, some are chart, some are plate, some are figures, something like that. So when you come to the last aspect, that is chapter 5. Chapter 5 is summary, conclusion, summary, and conclusion, and recommendations. So where we have chapter 6, normally some like maybe in PhD they want you to present your result alone, then you discuss another table, another chapter, then chapter 6 will be summary, conclusion, and recommendation. For example, in masters it will be summary, conclusion, and recommendation. So for summary, you find out, that is chapter 5, summary, conclusion, and recommendation. For summary, you try to summarize. Although some people are saying when you do abstract, then it's also part of the summary. But there is difference between abstract and the summary. Abstract is before you enter the work, people are giving you guide of what do you expect to see inside. But summary is when you finish reading, then they will try to summarize it for you so that you will now come out with a summary. So it's just like somebody who is presenting a paper. After presenting the paper, then you ask somebody to showcase his paper. What did he present? So that before people will ask questions, you now try to highlight what he has already just said. So summary, you summarize the work and you give contribution to knowledge in summary. You try to give contribution to knowledge. Maybe after trying to summarize in a shorter paragraph, so you try to highlight contribution you made in that knowledge you obtained. That is very important, contribution to knowledge. Then conclusion, you try to conclude. Okay, when you conclude, you also look at your objectives. You look at your objectives, then, but when you look into objective, you have to find out which one is the major objectives, which one is the concrete objective now. So for those concrete objectives, for those major objectives, that means you have to conclude on them. So try to come up with the conclusion based on your own objectives. You conclude based on the objective, and then you now make recommendation. Recommendation based on your findings, please. Based on your findings, try to look at your findings and come up with the recommendation. Some people will give general recommendation firstly, then they will follow with a concrete recommendation. Please, I advise you to start with a concrete recommendation based on your findings. What did your findings said? So from your finding, you have found out that so extract, maybe ethanolic extract, is extracting more of the constituent of this plant than the other plant, than the aqueous plant, than the aqueous extract. So you stated that you now said you try to recommend. So you are now recommending people to use methanol to extract so plant, because when you use ethanol, it is extracting more of the constituent, plant constituent, and therefore you have more active ingredient or constituent in that ethanol extract. So, but people will now begin to say, to generalize recommendations, say they are recommending for further work. How do you recommend for further work without giving your own findings? Give recommendation for it. That is what shows you have done the work, because you have to crack your brain and come up with the findings, recommendation from your findings. What do you find? And when you find this, then you think over it and recommend appropriately. That means before you begin to give recommendations, that it is recommended for public enlightenment, you are recommending for future work. All this recommendation will come after your own concrete findings from your findings, concrete recommendation from your findings. Then we have references. Normally here we use alpha reference, and try to punctuate all your references, punctuate very appropriately. You see, sometimes the more the reference is looking very fine, that shows you have done a work. That shows the work is very, very nice. It's just like somebody who used to wash his car, to polish his car, and somebody who doesn't wash his car. It's just like that. You know, the more you take care of your car, that shows how you take care of yourself. So the more you take care of your reference, that shows how you take care of the work, because you have dedicated your work and time, and you are very committed. You have to do all the punctuation, all the corrections, all the paragraphing in the reference. So the reference needs punctuations actually, and you have to utilize name of journal, name of textbook, utilize it. And then you have to make sure inside the one cited inside must appear in the list of reference. So what you do is, please take your time. When you see, begin from just chapter 1, just the one you see, tick it, make sure the date tallies, make sure the name is correct, make sure if there are two people, it's only two people, and there is also two people. Sometimes people will say somebody et al, and then when you go there, you just see it's only one person. So you have to take care of the date, check the date, check the name of the spelling of the people, check everything. Please make sure, so as you are ticking, go, you finish number 1, and then number 2 also go and check number 2 and go to the list and tick. You tick in the list of reference. When it's not there, you put not listed. Or when you finish listing, so all those that appear not tick is not cited. It means you didn't see them. So you have to go back and make sure you see them. So that is how you do a checklist. Then appendices. Appendices is something that is not your result, or is not a result, but it's something you want to show people, like a questionnaire, like an ethical approval. You have obtained ethical approval, just put it there in appendix. Maybe you have designed a questionnaire, put it in appendix. Maybe some ingredient used in the culture media, or maybe some preparation method of the culture media, or some statistical analysis that are very bulky. So you try to summarize it and put it in there, but you can extract some other things and use it as evidence you have conducted statistical analysis inside your work. But the main statistical work, you can put it in as appendix. Maybe some people use glossaries after appendix. Some people just stop at appendix. So please, these are in a nutshell how research is supposed to be reported after you have conducted your own research. This is how you write it step by step. Thank you very much. Till we meet next session. Thank you.